Hi everyone. This week's lab is a gene expression activity, and it's meant to be fun but also educational and have you practice your skills of doing transcription and translation. So a review and overview of this process of gene expression. So this is how genes are converted into proteins. So genes are instructions for building proteins. So our proteins, all of the proteins in our cells are encoded in our DNA. And the first step of this process is to make an RNA copy of the DNA. That's called transcription. And then to translate that RNA code into amino acid sequence to build the protein, and that's called translation. So those are the two steps of gene expression: transcription and translation. As a reminder, the DNA lives in the nucleus of the cell. So transcription happens in the nucleus. Then the RNA is sent out into the cytoplasm or into the endoplasmic reticulum, where it can find ribosomes, and the ribosomes read the RNA and convert it into the protein sequence. They are the ones responsible for doing the translation. So yeah, that's the process. How do I go to the next slide? Oh, there we go. Figured it out. Okay. So overview of nucleic acids. So the building blocks of DNA and RNA, the individual monomers, if you will, are called nucleotides. And this is the anatomy of a nucleotide, if you will. It has a sugar. It has an amine base, so a nitrogen-containing base group sticking off of it, and it has a single phosphate. All right, so the difference between RNA and DNA, we'll see on this slide in a second, but they both are made up of these monomers that have slight differences, and they link together to form polymers, and those linkages that connect them are called phosphodiester linkages. So remember, with monosaccharides, we linked them together with glycosidic linkages to make a polysaccharide. So a theme in this unit is these important biomolecules being made up of monomers, individual units that are then linked together in long chains that are called polymers. And so making sure you understand what the monomers of these different classes are and the polymers are called. So they link together from sugar to phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, as you can see. And so we call this part of the DNA or RNA the sugar phosphate backbone. Now DNA is double-stranded, and it forms like a ladder that's twisted. We call it the double helix. Okay, so there's two strands that go in opposite directions. So you can see this top one goes 3' to 5', and the bottom one goes 5' to 3'. So they're going in opposite directions like a highway. You've got like the northbound and the southbound lanes. And then the bases, this base group here, is what sticks out into the center, and it's those bases that form the rungs of the ladder. So the sugar phosphate backbone is the sides of the ladder, and the bases pair with each other to form the rungs or the steps of the ladder. So you can see here that there's these sort of red dotted lines connecting the bases on the opposite strands of DNA. And those red dotted lines represent hydrogen bonds that are holding those base pairs together. And you'll notice that A and T always base pair and have 2 hydrogen bonds connecting them, and G and C always base pair and have 3 hydrogen bonds connecting them. So a little look at those differences between the different nucleotides. So we've got DNA deoxyribonucleic acid and RNA ribonucleic acid. So one of the ways that they are different is in their sugars. DNA has deoxyribose, and RNA has ribose. Literally, deoxyribose means ribose without an oxygen. It's missing an oxygen right here. So this one has an OH and this one has an H. Otherwise, the sugars are completely identical. Another way that they differ is in their bases. So DNA has A, T, G, and C, while RNA has A, U, G, and C. So the T is DNA only and the U is RNA only. A third difference is in the number of strands. So DNA is usually double-stranded, RNA is usually single-stranded. There are exceptions for both, but and then I guess the other would be, a fourth one could be their location in the cell. So DNA is in the nucleus and RNA is made in the nucleus, but then it gets transported to the cytoplasm. All right, the base pairing rules, again, to remind you, A pairs with T if it's DNA, it pairs with U if it's RNA, and the mnemonic for that is apples in the tree. A pairs with T, and G pairs with C. The mnemonic is the car goes in the garage. So a little animated video here that we're going to watch of this process, because the thing is that transcription and translation are very dynamic processes. It's hard to visualize them or to conceptualize them when it's just a picture. So watching the animated version, I think, is better. So we've got a cell. Turn on the volume.
The nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
The end. So that's the process from start to finish. All right. So in this lab, you are going to be doing this process. And so this is sort of an overview of those different things. So you have some double-stranded DNA on top here, and the DNA has A, T's, G's, and C's coding. So there's 2 strands. There's the coding strand and there's the template strand. The template strand is the one that RNA polymerase uses to make a complementary strand here of the coding strand. So the mRNA is basically a copy. It ends up being a copy of the coding strand. So let's see how that happens. So let's do the transcription. Here's my marker. Can I draw on here? Yes, I think I can. Okay, so we're using the template strand here. So this is our DNA sequence, and we're going to write the complementary mRNA sequence. So T pairs with apples in the tree. So A pairs with T. So the complement of T is A. The complement of A is T, except this is RNA. So in that case, it's U, because RNA uses U, uracil, not T-thymine. My daughter's mnemonic is that unicorns eat apples. So that's how you can remember. And then C pairs with G. And then we'll do it again. G, the complementary base is C. A pairs with T. C pairs with G. T. Oops, I made a mistake. I put a T here, and that's okay if you do that. You'll probably be reminded when you're using in the next step, when there's no T's in this table, that all of your T's need to become U's for the RNA. All right, T pairs with A, G pairs with C, A pairs with U in the RNA, T goes with A, G goes with C, and C goes with G. All right, so what we just did was transcription. We took the DNA sequence and we transcribed it, we converted it to the complementary RNA sequence. So now we have made an mRNA transcript. That will leave the nucleus, it'll go find a ribosome, and the ribosome will read these triplets, which we call codons, and translate those codons using this table here, the code on this table to predict the amino acid. So let's start with the first one. We've got AUG. And we look at this table and we see AUG is right here. AUG is the start codon. Another table will tell you, the one in the lab will also tell you that that also means methionine. So this is the start, and it codes for methionine. I'm going to try to draw it going this way. The three-letter code for methionine is MET. All right, now we're going to the next codon, CUG. I look on the table and I find CUG right here. That codes for leucine. So this is L-E-U, leucine. ACU. ACU is right here, and that codes for threonine. And ACG is right here in the same box, also codes for threonine. So you can see how there's some redundancy in the genetic code. Different codons can still code for the same amino acid, all right? That third position, so you notice that like AUU, AUC, AUA, all code for the same amino acid, so that third position is, we say it's wobbly, that there's wobble, because a lot of times that third letter can change and you still get the same amino acid. That is really important in our genetic code. It means that we have a lot of redundancy in the coding, which allows for the mistakes to sometimes happen, but be sort of like typos that don't matter. You could still read the word. You can still make out, it'll still make sense. The protein sequence doesn't change. We call that a silent mutation, okay? So what we just did here, going from the RNA code, the RNA codons, the three-letter codons, we went to the amino acid code, and that's called translation, and that happens at the ribosome. Translation happens at the ribosome. That is the enzyme that performs this job. Transcription is performed by RNA polymerase. That's the enzyme. Just polymerase. It makes a polymer of RNA. That's what it means. Okay, so in this lab, how do I unclick my pencil? Oh, there we go. Okay. Here's the lab here, the gene expression of a mystery organism. And so this is supposed to be fun and creative. Okay, so on this page here, page 2 of the lab, you have your sort of coding references. All right, so here is a codon table that's very similar to the one in the slide. All right, and it tells you the 3-letter amino acids, and then it also gives you over here what the full name is and what the 1-letter code for those amino acids are. And then up here, this is giving you various amino acid sequences and what trait they code for. So proteins give rise to traits. And so this is a very oversimplified activity because proteins tend to be like 50 amino acids or longer, and we're just making up some proteins that are 2, 3, or 4 amino acids for simplicity. And a lot of traits, human traits, are complex and are due to multiple proteins working together, and we are pretending in this simple organism that a single protein is a single trait. Okay, and so you'll refer to this table. So you will have a mystery creature genome, all right, and it has several genes. So there's 6 genes, A, B, C, D, E, and F, that are already here for you. So it gives you the DNA sequence. Assume this is the DNA template strand. So your job is to do transcription and come up with the mRNA sequence. Then do translation and come up with the amino acid sequence, and then refer to that table to see what trait that gives rise to. So you're going to decode these 6 different traits, all right? And then you're going to reverse engineer 2 traits. So you'll pick 2 other traits from the table. So let's say you were picking pointy horns, all right? So you need to, so you'd start instead with the amino acid sequence, glu-tyre, glutamate, and tyrosine. So you'd start with the trait, the horns, then you would write out from the table what the amino acid sequence is, glu-tyre, whatever it was. Then you go to the codon table and you look up what could be the codons, 2 codons that code for each of those amino acids, and then what would be, you do the reverse of transcription, you write out what the complementary DNA sequence would be. So you're doing this process in reverse for 2 additional traits. So then at the end, using colored pencils or markers, or if you're on a tablet, you can do it, I don't care, do watercolors, whatever you want. Draw your mystery organism. So draw an organism that has these 8 different traits. And there's definitely creative license there. Even if 2 students have the same traits, their organism will probably, the picture they draw will probably look a little different. And that's true of organisms as well. You can have identical twins that have the exact same genetics, but they still look a little bit different. They still have different personalities because environmental conditions also contribute to how those traits present. So that's one of the reasons I like this activity, because even identical sets of traits can result in 2 very different looking presenting organisms. So that is the lab activity to practice this transcription, translation, and form a creature from that. If you have questions, you know how to reach me.